lecture, your A&P career, academics, or whatever. Not that you won't be using it. Last time, I have to check on the Tuesday, Thursday schedule, but the Monday, Wednesday schedule had the digestive lab report due the same day that we were doing the lab on Wednesday. So I don't know if it says the same thing for you guys, but if it does, it's not due on Thursday. That's the day we're doing it. And the report will be due Tuesday at 5 on the day you take your exam. Okay? So we are doing a wet lab on Thursday. So we're going to be looking at the digestion of fat and whipping cream. So that means it's a day for goggles and closed-toed shoes. All right. I bought some whipping cream today, actually heavy cream. I don't know the difference. It's heavy cream and whipping cream.
One's light.
One said whipping cream, heavy cream, and the other said, one said whipping cream, this is heavy whipping cream. Anyway, they're both lipid. All right. So we're going to be looking at the addition of pancreatic enzymes, which is pancreatic lipase. And this is digestion that occurs in the small intestine. So what's the pH going to be, or should be, the digestion that occurs in the small intestine? Anybody know? What's the pH of digestion that occurs in the stomach? 1 to 2. Okay. We're going to take our physiology of digestion through the stomach and begin control of small intestine, but we'll finish that up on Thursday as well as looking at hormones. All right. Interestingly, digestion that occurs in the mouth is slightly acidic, 6.5 to neutral. Digestion that occurs in the stomach is very acidic, with the hydrochloric acid around a pH of 1 and 2. However, those enzymes no longer work in the small intestine. The pH is modified to an alkaline pH of 8 or 9. And so the cream, the enzymes from the pancreas need to have an alkaline pH of 8 or 9. So when you see the cream on Thursday, it's going to have a purple, lavender, light purple color, indicating that's the litmus powder that's been mixed up and swirled into it. That's our litmus or pH indicator. So that lavender purple color indicates an alkaline pH. It's going to change, if digestion occurs, it's going to change to a mauve, kind of a reddish purple or pink color. And that shows that the pH has decreased to an acidic pH. Not because of anything acidic that we're adding to it, however. What would be the products of lipid digestion? I know we haven't gotten to it yet, but what do you think would be the products of lipid digestion that make it an acidic pH? What are fats made up of? We're looking at triglycerides, by the way, not cholesterol. And we're breaking down triglycerides. We have glycerol, which is the 3-carbon. Anybody know what's added to the glycerol? When we look at the digestion towards the end of lecture today, let me switch to that. So here's glycerol. And these structures here, lots of carbons and COOH is our fatty acid. So when we break down the fat in the whipping cream on Thursday, using pancreatic lipase, some of our products are going to be fatty acids. And that will lower the pH of the cream and change the color from the lavender to that mauve or reddish pink, purplish pink, right? So that's one of the major characteristics that we're going to look at to determine if digestion occurs. So we'll have that wet lab, plus we'll look at on PhysioWet the digestion of carbohydrates and protein. Okay? So this is where we're going to end up tonight, but let's finish our anatomy of the colon or rectum, and then we'll look at pancreas and liver and pancreas in that order, and then we'll take a break. Let me study for your quiz. Take the anatomy quiz over structures through, but not including, up to, but not including the colon and pancreas and liver we're talking about tonight. Okay? And then after you study for that, we'll take our quiz, we'll come back and do about half of our physiology left. The other half we'll do on Thursday. Alrighty, so giving everybody an opportunity to look at this first question. Remember either only serous or primarily mucus or a mixture of serous and mucus, okay? So what are you seeing here? A mixture or primarily one or the other? So what type of a secretion is this? Mucus, and this? Serous, okay? So this would be a mixture. If it was primarily mucus, it would be almost all white with just some of these little, what they're called serous dimilutes. And if it was primarily serous, it would be all of this kind without the mucus. All right, so this one is a mixture. So which of those salivary glands is a mixture of both serous and mucus, although it's primarily serous? Has everybody had a chance to answer this? All right. So we're looking at the colon. And let's look at the growth structure of the colon, and we'll come back and look at the GPAT or the histology of it. So, small intestine, the last portion of the small intestine is the ileum. And it ends by entering the large intestine at a blind pouch region known as the cecum. All right? And there's another sphincter here called the ileocecal sphincter or valve. So far we've had the orbicularis oris, which is a sphincter of the mouth, which is a voluntary muscle. A sphincter between the pharynx and the esophagus that I've mentioned, but you don't have to know. Then the smooth muscle sphincter between the esophagus and the stomach, the gastroesophageal, or the cardiac, right? Then between the small intestine and between the stomach and the small intestine, we have the pyloric, another increase in smooth muscle. And then now between the ileum and the large intestine, we have the ileocecal. So this is the appendix. We talked about this a little bit with lymphatic system. Its complete name is the vermiform, which means worm-like. And the appendix actually has its own mesentery. See that here? Although it's difficult sometimes to see the appendix because it's usually tucked up behind the back of the cecum. All right? So this region is the cecum. And then we have these pouches or haustra of the colon. Haustrum is plural. I mean, haustrum is singular. Haustra is plural. And they vary from species to species, all right, as does the movement. So cows and elephants tend to have a more rapid movement and larger pouches, so they make the patties, if you will. Their bowel movements are, stools are soft and with all the grassy and herbivores to show that they eat. Other animals have dark, small, hard pellets. So that has to do somewhat with the haustrum and the speed with which the food moves through that. And helping to create these are 3 strands of smooth muscle. Depending on the angle at which you see these, you may see 2. You won't see all 3 at one time, and these are called tinea coli. In fact, those are the remnants of the outer longitudinal layer of smooth muscle. Okay? So there's only 3 of them, so instead of completely surrounding the colon, they're limited to these 3 bands. Then we have a hepatic flexure or right colic flexure. You don't need to know that, it's just called for where it bends. And the splenic flexure is actually higher because there's more room near the spleen. And then we descend for the left descending colon. So this is going to would be the ascending colon, the transverse colon, and remember that's in the mesentery. So what's the name of this mesenteric structure that suspends the transverse colon? Gave you a clue. Transverse mesocolon. So you'll notice I did diagram a mesentery for the ascending colon. That's because the ascending colon is a retroperitoneal structure. Okay. The transverse colon, because it has a mesentery, you know that it's intraperitoneal. And then we have the descending colon, which is retro again. Then we have, I need to draw this just to give me a way to plan to put it in. All right, so we have the sigmoid colon, and that has its own mesentery, the sigmoid mesocolon, which is not on your lab list, but it does allow us to say that the sigmoid colon is intra. All right, and then we have the rectum, which means straight. We come down straight, and we do the anal canal. So how do we describe the rectum, the placement of the rectum? Is it intra or retro or something else? Remember the word for below? Infra. Okay, so this would be infra. And we have our last two sphincters, a thickening of smooth muscle. That is the internal anal. And then part of our levator ani, which is skeletal muscle, and is the external anal sphincter. So for those of you in the back that can't see this far down, internal is slightly superior, external, I'll draw it up here. Here we have our rectum and anal canal. That would be the internal anal sphincter. And then we have the external, which is the region of the levator ani. So this would be rectum. And anal canal. Okay, so let's do a review of the relationships to the peritoneum, of the structures that we've covered so far, before we look at the histology or the GPAT of the colon. And I'm going to leave that up just a little bit longer, but I'm going to erase the diagram in the upper corner there for those of you in the back. Of course, the mouth has nothing to do with the peritoneum, so it obviously is not going to be anything in relationship to the peritoneum. What would the esophagus relationship be to the peritoneum? Most of the esophagus is in the neck and the thoracic cavity. But there's a small region of it, about an inch or so, an inch and a half or so, that does enter into the abdominal cavity. And that small inch and a half is retro. All right, so retro for about 1 to 2 inches. Okay. Then we have the stomach. Okay? So we have both the lesser omentum and the greater omentum for the stomach, so that would be intra. Small intestine, we have 3 parts. So let's look at the duodenum. What position is the duodenum? Retro, pushed over to the side, and the posterior aspect of the peritoneum is reabsorbed, so that's considered to be retro. And the jejunum and ileum are both intra, because they're associated with the mesentery proper. And then as you can see here, we have the cecum and ascending colon are retro. Transverse colon is intra. And I'm running out of room here. Sigmoid colon, sorry, descending colon would be next. That would be retro again, because it doesn't have a mesentery. And then sigmoid colon would be intra, it has a mesentery. And I ran out of room for the rectum. What would the rectum be? Infra. Okay? So the rectum is going to be infra, below the peritoneum. All right?
So does it always stand true that if it has a mesentery,
It's intra.
and if it doesn't, it's retro? Okay.
Yeah. Or infra, as the rectum is. Okay? All right, so let's look at the histology of the colon. Here's a review of the blood supply. So if you recall, the superior mesenteric artery does all of the small intestine. It also does the ascending colon and transverse colon. It stops here at the left side, where the ligament flexure is, and the inferior mesenteric artery picks up. There's a little bit of overlap right about here. What also stops at the distal end of the transverse colon is the innervation by the vagus nerve. The vagus nerve is going to innervate liver and pancreas and stomach and small intestine, ascending and transverse colon. And then for parasympathetic innervation, it picks up with the pelvic splanchnics. Okay? And then the sacrosplanchnics who do this descending colon. The rest of it is done by splanchnics from the sympathetic chain above the diaphragm. All right? So here's a view. We can see the muscularis mucosa. So everything from up here is mucosa. Here's our submucosa. And then we're seeing part of the muscularis externa. We're not seeing the serosa here. So may I erase the board? And we'll diagram that portion up here. So if you recall the basic pattern of the mucosa that I drew on the board last Thursday, I'll redraw it. So we had the gastric pits for the stomach. And gastric glands. And a muscularis mucosa. Very smooth surface. All right, and then the pockets with the gastric pits. The small intestine. To increase surface area, we added villi. We still have glands, so we have our intestinal glands. So this one's the more the muscularis mucosa. Okay? So the mucosa is a simple columnar epithelia with about 25% goblet cells, which is what creates most of this white appearance. Looks like it's more than 25%. What do you think the mucus from these goblet cells, what is the purpose for that? Lubrication. Our pH has been brought back up to around 8 or 9, so it's on the alkaline side, but it's not like 10 or 14. It's not as acidic as the 1 or 2 of the stomach. We have a loose irregular connective tissue. And we have muscularis mucosa. Okay. The submucosa is standard, so just a dense irregular connective tissue, nothing unique there. And we do have some modification with our muscularis externa. We don't have the inner circular layer. And of course, this is smooth muscle. And the modification is in the outer longitudinal, which, as I showed on the board, is limited to the 3 bands. And then we have a serosa, but only, of course, on the anterior part if it's retroperitoneal, and completely around it if it's intraperitoneal. And we still have the serosa, which is our visceral peritoneum, same thing, loose irregular connective tissue. And simple squamous epithelia.
How far around it, what's it from the retro?
Pardon? It's only over, so I can find the ascending colon, it would just be, you'd have parietal peritoneum and go over the front, down the sides, and then on to the posterior abdominal muscle wall. So, just enough to anchor it to the posterior abdominal wall. Alright, so here's a closer up view of the submucosa on the mucosa. Okay. So last night I told the class a story about a former patient of mine that had his mucosa totally destroyed when he was 5 years old. He grew up down near the Grapevine, near a bunch of farms, commercial farms, and they were growing lima beans. He liked to go out and eat the fresh lima beans. Well, he didn't realize one day, being only 5, that they had been sprayed with pesticide. And his esophagus and the stomach and small intestine recovered, but for some reason, it just chemically burned out the large intestine, the rectum. And not only did it remove the mucosa, but it left adhesions and scar tissue. So he was no longer able to have a regular bowel movement. Instead, he had to have high colonic enemas. He never had a colostomy. He was happy he didn't have to do that. So the rubber tube was placed up in this region. So I met him when he was 81. 81 or 82. And his whole life, that was how he had a bowel movement after that. He went to vet school, worked as a vet, taking care of the army horses in Asia during World War II. And even though he was 82, he was up here. His home was down south, and he would work up here in the Sacramento area. And so he hired me as an RN to give him these chronic enemas. So most people go to the bathroom every 1 to 2 or 3 days. You don't feel very good if you go more than 2 or 3 days without having a bowel movement. He would sometimes go 3 weeks. And he got really, really busy. I saw him about once a week if he kept on schedule. In his house in Southern Valley, he had a special bathroom built. He had an RN come there. And I remember the very first couple of times I asked him why he didn't have his wife do this, because she would always be puttering in the back of the house when we were in the front bedroom. And he said, she's my wife, she's not my caregiver. In fact, he hadn't even told his 2 daughters that he had this condition. So first time I went over there, I got there about 6:30, and I didn't leave till after 11. And so he had an apparatus, he had an old-fashioned IV stand like that Coke rack over there, and a glass jar that resembled a blender. And the tubing was latex about the diameter of my thumb, but stretched out over the bottom part of the blender, which held about 2 quarts. And so we start out with just plain tap water, about 2 quarts of that, and I lubricate the tubing and put it into here. He thought I did a good job because I was an anatomist, but I just went slow. And then I undo just a simple clamp and repeat that until he'd taken about 8 to 10 quarts of fluid. I'd see his toes start to curl and encourage him to take a little bit more, and then he'd say, okay, that's enough. And he'd go into the bathroom right off the bedroom, and I'd sit on the couch and read my research papers and did read his work papers for about 15, 20 minutes. And then he'd come back and we'd do it again. So we do it 3 processes. The first was tap water, the second was soap suds, just wash my hands with ivory soap, and then the last one was saline. Add some salt, table salt to it. And so his son-in-law came one summer, the second summer, I did it for 2 years, and he was concerned about how we were going to do this without his son-in-law finding out. I said, he doesn't know you have this condition? He said, no, I never told any of my kids about it. Yet he'd gone through his entire life with this condition. And that was the only way he could get his fecal matter past all of this area. But he was so grateful he hadn't had to have a colostomy. Colostomy is temporary. Someone has ulcerative colitis. They may, to allow the colon to rest, bring one region to the surface from opening or ostomy in the skin, and suture it to the skin. And then there's a device, an application device, that uses special types of skin glue around that, and then you can just add and replace the colostomy bag. Now, if it's over in this area, which is very close to where it's coming from the ileum, it's very liquid and gaseous and foul smelling. No matter how much room refreshener you use, it can't mask that smell. If it's over in this area, it's usually solid, and they can oftentimes, not every time, but they can oftentimes train themselves to have a bowel movement, a regular bowel movement, into the bag. All right. It might be, if it's colon cancer, then it's going to be permanent, depending on where the location is. If they remove too much that they can't bring together, and there's not enough room for water reabsorption. However, he wasn't the only one in his family that had to have that done. His aunt, when he was born, was already having this because she had lost the lower region, the sacral region of her spinal cord, which is parasympathetic. So parasympathetic is going to be doing peristalsis of the colon and relaxing the internal anal sphincter to allow for a bowel movement. So she was dating this guy, and they were supposed to go to the fire station for a community dance, and he was about 45 minutes late. And her friend said, well, he knows where the dance is, so just go ahead and come with us, and he can meet you there. So that's what happened. He showed up, and while they were dancing, he shot her in the lower back with a small pistol. And she became paralyzed with her lower limbs and lost the parasympathetic function. And so he took care of her when he was older. Would take her on trips up and down the coast, carry her in and out of the but it was a help to him to not be the only one that had to have these colonic enemas. So he had sleep apnea, and he died a year or two later with heart condition from this, but from 5 to about 83 or 84, he lived with this condition. And it was just, like I said, it was just a good illustration of how important all the mucus secretion is for the lubrication. So the large colon, as we'll see on Thursday when we talk about finished physiology, talking about absorption, is where water is primarily absorbed, all right, along with some vitamins, especially vitamin K, that's produced by the bacteria that normally live in the large intestine. All right, when we get to the anal canal, the last couple of inches there, this is just an illustration showing how the posterior aspect, so this is flipped around, the appendix, the posterior tena coli can lead you right to the appendix. The surgeons will use that if they have a hard time finding that. Which, talking about, you guys know my daughter-in-law last January, February, had in spring, had the gallbladder problem issues. So I normally pick up my granddaughter on Tuesdays, and I always check, because sometimes she gets off early and brings her to the grandparents. I texted my son this morning and asked whether or not I needed to pick her up, and he said, no, you can come play with her though, and I said, I'm still feeling sick, and he said, well, everybody else is sick. Michelle was in the ER last night. So I said, what? And he said, so she's 25, and he said, a little hernia. And that's what babies get. Prior to it, we talked about the longitudinal folding, the linea alba between the rectus abdominis. Well, adults can get that too. Weight has a factor with it. Multiple pregnancies. She's only had one. But severe abdominal pain is one of the signs. And surgery, I said, well, does she need to have surgery? He said, I don't think so. The doctor just told her what she had and sent her home. Well, from what I read about it, it practically always requires surgery in the adult. In the baby, it can heal on itself, but in the adult, it doesn't. It's an in and out surgery, but the loop of intestine shows up as a bulge right at the belly button. Of course, that can get caught and twisted. So, one more issue. But when we get to the anal canal, we're in the last 2 inches here. All right, I'm not going to diagram the mucosa anymore, but we do switch back to our original epithelia, which was what? What do we have in the mouth, the esophagus? Non-cornified stratified squamous. So that's what we have in the anal canal, again, for the abrasion of the more dry fecal material. If you recall, we have the internal hemorrhoids and the external hemorrhoids, which if the fecal matter is very dry, can cause bleeding and tearing of that. So here would be the internal anal sphincter, and out here would be the external, the levator ani. Anybody remember what the blood vessels are, the veins, that if they lose their valve continuity can result in the internal hemorrhoids? Middle rectal. So middle rectal veins are where we get the internal hemorrhoids, and inferior rectal veins are the external hemorrhoids. Okay? Okay, so question 2, identify the nerves which results in relaxation of the internal anal sphincter. This is what allows a bowel movement. Of course, the external has to relax too, but for someone who's not potty trained, this is the major one. So is defecation a normal homeostatic event, or is it a fight or flight response? That's the question you need to answer to figure out which of the nerves. I don't know, if you're running away from danger, it might help you get away to do stuff behind you. So the answer is actually, well, the clue is on the board, on the screen. The answer specifically is not. What's the clue? So parasympathetic does peristalsis throughout the digestive system. But what does it do to sphincters? During contraction of the smooth muscle for peristalsis, what's happening to the sphincter? Think back to the bladder. detrusor contracted, the internal sphincter relaxed. Same thing occurs with the digestive system. The muscles contract for peristalsis, but the sphincters relax. Okay? Sympathetic does the opposite. The smooth muscle for peristalsis decreases their stop movement, and the sphincters constrict.
Which nerve is parasympathetic? Again.
You ready to move on? Still thinking about it? So I showed you this slide on Thursday night. We talked about two junctions where we change epithelium. First one was the cardiac sphincter, where we change from the non-cornified stratified squamous of the esophagus to the simple columnar of the stomach. And the second one is the anal rectal sphincter, where we change from the simple columnar of the rectum to the non-cornified stratified squamous of the anal canal. So that's what this one is, all right? And the key factor is the amount of goblet cells that we see in the intestinal glands. The entire gland looks like it's full of goblet cells, whereas in the stomach we have the mucus cells just up in the pit area. So it's a distinctive difference in the appearance, plus a thicker stratified squamous region. All right, so let's switch over now, and I'm going to change slides, and we'll look at the liver and the pancreas.
How did you feel on the test? I got 65. Did it lower your grade?
All right, so the liver appears here to have two lobes. From the front, it does. So we have a large right lobe and a smaller left lobe. And then if we switch it over to the back, we can see those two lobes, but in addition, we have two smaller lobes. You can see them here. We have the quadrate lobe and the caudate lobe. All right, anybody know what this structure is right here? You can see it on the side of my head here from the screen. So the umbilical vein, the collapsed umbilical vein. Anybody know, remember what we call that if you're looking at the screen? Ligamentum teres. And on the opposite side of that lobe, kind of blocking it off, we have the ligamentum teres on one side, and then we have the gallbladder, kind of framing it on the other side. And then more at right angles, but it's kind of hard to draw it that way. Up here we have the collapsed ductus venosus draining into the inferior vena cava, so this would be the ligamentum. Venosum, and we're seeing part of the inferior vena cava. And so this lobe here, all right? Right in here is where we're gonna find what's called the porta hepatis, although you don't have to know it's for the hepatic artery, and the portal vein, both care empty into the liver. So this lobe here is identified as the quadrate. It has 4 sides, a quadrangle. So this is the quadrate lobe. This would be our right, and this would be the left. This one also has 4 sides, but we can't have 2 quadrate lobes. So this is on actually the posterior aspect of the liver, and another, which is where animals' tails are, and another word for tail is caudal. So this is the caudate lobe. Okay, let me bring over a couple of our models and illustrate that. So remember this weird one where the stand is really on the front of the liver? So this is my liver, I'd be very sick. Okay, the liver should not be nearly this large. In fact, you shouldn't be able to feel more than a half to maybe an inch of the liver below your ribs, certainly not more than an inch. And I don't have that much room under my diaphragm for it. So this would be the right lobe and the left lobe. And we turn it around. Here would be the ligamentum teres, okay? And here's the gallbladder. So this lobe right here matches the structure here. We have ligamentum teres and the gallbladder, and this would be the quadrate lobe. Then if I turn this slightly, you can see the ligamentum venosum and the inferior vena cava. So the liver wraps partially, sometimes even completely around the inferior vena cava just before it passes through the diaphragm. And this would be the caudate lobe. So you can see that it really is in posterior, on the posterior aspect. Now, I didn't mention it when we were talking about the ligamentum teres, but we actually have one more mesentery to talk about. And that is a mesentery, a double layer of peritoneum that suspends the ligamentum teres. So if we look at this is a transverse section, and we look at the anterior abdominal wall, looking from the top down here. Here would be our ligamentum teres, which if you remember starts at the belly button, umbilicus, and moves up to the liver where the umbilical vein is coming in. And if this is our peritoneum, this would be parietal peritoneum. So it suspends the ligamentum teres from the region of the umbilicus up to the liver. Okay? And that would be the falciform ligament. If we look at it, so this would be a transverse view. If we look at a more sagittal view, so cutting the length, and here would be prior to cutting the umbilical cord. Here would be the umbilical vein coming in from the placenta. This would be the belly button, and traveling up to the liver. Then the falciform ligament is going to be found along this region here, kind of suspending just that portion of the umbilical vein. Okay. So falciform ligament there and here. You can see a little bit of it right here. This is a cadaver liver. From the front, right here, and then the passage between the right and left lobes there. And you can see a little bit in this diagram here. So there would be the ligamentum teres, and that would be the falciform ligament where it's been cut in the abdominal wall. So in this diagram, here's the gallbladder. So in this area here would be our quadrate lobe. And we have the bile ducts that are shown here kind of as if they're looking through the liver, okay? So the liver has lots of functions. It produces most of the proteins that we find in the blood. We've talked about the fatty proteins. We've talked about complement. The liver makes those. It produces the major osmotic protein or albumin. All right, so any liver diseases are going to not allow those particular tissues, those particular proteins to be produced. So what type of cells is the liver formed of? We have about 4 tissues: epithelial, connective, muscle, and nerve. Which of those 4 is going to form the functional cells of the liver? The liver is classified as a gland. So what kind of tissue forms our glands? Epithelial. So in 4:30 we talked about the fact that epithelial tissue is lining, like lining the mouth or lining the stomach, covering our skin, our body as the skin, covering our organs as the peritoneum or the pleura, and as glands. All right. So here we're seeing it as a gland, and part of its formation, as we'll see in a few moments, not dumping it into the blood, but dumping it into these ducts, is the formation of bile. And bile salts, as we'll see on Thursday, help to maintain an increase in surface area. So if you were to add, if you're making salad dressing, and you were to take olive oil and vinegar and add them together, they're not going to be miscible. They're not going to mix very well. All right? So if you shake it up, that shaking, that vigorous mechanical movement of shaking is going to break down the olive oil into smaller drops and disperse it amongst the vinegar. So you'll have this kind of foamy, cloudy solution. But if you let it sit, they're going to separate. The oil droplets will find each other and coalesce, and you'll have a separation with the lighter oil on the top and the vinegar on the bottom, which is water as well. So what happens to surface area when all those little droplets come back together to form a layer of oil? It decreases. We just have that surface interface between the water and the oil. If there were any enzymes in the vinegar to work with the oil, it would be just that single surface. So with that mechanical churning that occurs in the mouth, that helps to mechanically break down the lipids in our food into small little droplets. Not chemically change them, they're still small little droplets of fat. But they would recoalesce into larger droplets and decrease surface area. And so one of the secretions of the liver are these bile salts that attach themselves, as we'll see on Thursday, to the lipid droplets and keep them from recoalescing, kind of like opposite charges. So when they come back together, they're repelled, all right? So the bile then is added along with the acidic chyme and our enzymes from the pancreas is added in the region of the duodenum. So let's look at the pattern for the duct structure. And we have tiny, tiny, tiny little, I'll leave it on that one, I have another diagram. We have tiny, tiny little ducts within the liver, and then they come together to form larger ones. So may I erase the board? And we'll diagram and label those. Okay? So coming from the right lobe, we have the right hepatic duct, and from the left, we have the left hepatic duct. And the liver is constantly making the bile with the bile salts, okay? So as it's made, it drains down into these ducts. The right hepatic duct and the left hepatic duct join together to form a common hepatic duct. join with the pancreatic duct, but there is a sphincter here. We'll come back and label that in just a moment when we talk about the pancreas. So we're talking about this area right here. And that sphincter is going to be closed unless there's a lot of lipid in the food that you're eating, all right? So what happens then is the bile backs up and it flows into a cystic duct. Cystic means bladder. It flows into the cystic duct and then is stored within the gallbladder. I don't know if you remember seeing the gallbladder when we did the sheep pluck, but it has a pretty thin wall, but there's still smooth muscle there, all right? So when you do have a fatty diet, that gallbladder will contract and add to the bile that's already draining into the duodenum. And so the cystic duct plus the common hepatic duct then together form the common bile duct. So this other duct up here is from the pancreas, and I'll come back and talk about that when we're done talking about liver tissue. Okay. So let's look at these epithelial cells of the liver. And you can see we have a six-sided structure here, which is known as a lobule. So the lobes are the large structures, the right lobe and left lobe. And then microscopically, we have these lobules, right? So somehow for me, it's easier to think of a stack of bicycle wheels with their spokes all stacked up on top of each other, being six-sided instead of round, all right? And the spokes coming out to the sides here would be these channels of very large capillaries. So here's another view of a lobule, all right? So running up the six corners of this lobule, we have branches of the hepatic artery, branches of the hepatic portal vein at each of these corners, all right? So if I could kind of draw this on here while still leaving the duct stuff here. So here would be a branch of the portal vein. And it would send horizontal branches up along the sides of that lobule. And it would meet another branch of the portal vein. And that would send branches up this side, where they would meet another portal vein. Okay, and three more over here. So this would be a branch of a hepatic, let's put it down here where I have more room. Branch of the hepatic portal vein. And then as you can see, right next to it would be a branch of the hepatic artery proper. So this would be formed by the union of these two vessels traveling to the center of this lobule. These vessels are known as sinusoids. The sinusoid is a large capillary. The capillaries are typically the diameter slightly larger than a red blood cell. So the red blood cells have to line up in single file, and that slows them down so oxygen has a chance to diffuse off and move into the tissue. So these are larger than that, but they're still just a simple squamous epithelium. All right. And so the flow is going to be from the combination of these vessels toward the central vessel, which is known as a central vein. And that would be this structure here. Okay. So then all the central veins from all the small lobules in the liver come together, and they flow to form the hepatic veins. So in our discussion with veins on the cardiovascular system, we talked about the portal hepatic vein coming to the liver, and then from the top of the liver, the hepatic veins empty into the inferior vena cava. Okay. So let me shorten up the pancreatic duct here, and I'll draw this back in again. And let's list in sequence the blood flow from our capillaries in the villi. So we have, of the small intestine, we have the villi capillaries. Those are going to join together to form what vein, carrying it towards the liver? What's the vein that leaves the small intestine? What's the artery that supplies the small intestine? Not inferior, superior mesenteric artery. So these capillaries are going to join together to form the superior mesenteric vein, which joins with the splenic vein to form from our pattern. Here's the superior mesenteric vein, here's the splenic vein. What's this vessel? The hepatic portal vein. All right? So from the superior mesenteric vein, the blood is going to flow into the hepatic portal vein, which is going to give off these branches. So from the branches of the hepatic portal vein, it's now going to flow into the sinusoids. And then the sinusoids come back together again and form the central veins. And then the central veins come together and form form the hepatic veins. And then hepatic veins drain into the inferior vena cava. So how many sets of capillaries do we have in that sequence? This is our typical cardiovascular sequence where we go from arteries to arterioles to capillaries to veins to venules to veins to the heart. Or do we have more than one capillary bed here? The capillaries in the villi of the small intestine form our cap bed 1. The sinusoids form our cap bed 2. So this is our second, we've already talked about a little bit with the cardiovascular system. This is our second portal system. Well, third portal system, our second venous portal system. The first was in the brain between the hypothalamus and the pituitary. Remember, we had the capillaries up in the hypothalamus, and then we had the portal veins, and we had the capillaries in the anterior pituitary. All right. So why do we have two sets? Why do we have a portal system here? In the kidneys, we had it to control blood pressure, right? We had two sets of arterioles between our capillaries. Here, we have it to bypass the liver. So as we absorb our nutrients, and here it's going to be amino acids and simple sugars, monosaccharides, are going to enter into our cardiovascular system. Where do the lipids go? You guys remember? Into the lymphatic system, into the lacteals, all right? So the lipids are going to come in via the hepatic artery because our lacteals are going to empty into the subclavian vein via the thoracic duct and go to the heart. Our hepatic artery is bringing in oxygen and fats, whereas our portal vein is bringing in simple sugars and amino acids. And the liver is the largest gland in the body other than the skin, but inside the body it's the largest gland. And it uses the amino acids to form all those proteins that we just talked about. Okay, complement and clotting proteins and albumin for osmosis. It stores our excess sugars under the influence of insulin so that we have a quick and ready source. If we haven't eaten for 4 or 5 hours, it's going to start being released from the liver even 2 hours. All right? So it's going to go from the digestive system to the liver before it goes to the heart to get distributed to the rest of the body. Same thing that happens in the fetus. So from the placenta, the umbilical vein, which is rich in nutrients, goes to the liver first. All right? And although some of it will bypass through the ductus venosus, most of it will, it still goes to the liver first. So the liver gets first dibs before any other organ. Right? So we have the branches of the portal vein, branches of the hepatic artery, and that mixes and is carried within the sinusoids. You'll notice from this diagram that the hepatocytes, that's what we call those round cells, are never more than 2 cells thick. So think of this as a plate of cells, and remember I talked about stacking up the bicycle wheels, and think of the spokes, a big line on each side by cells, and never more than 2 cells thick, because between the backs of each cell runs a small channel into which bile is released, the bile salts that we were talking about. That doesn't go into the blood. That goes in the opposite direction and empties into small bile canaliculi. So between the hepatocytes run these tiny little bile canaliculi, and they empty into bile ducts, which are formed of simple cuboidal epithelia. So the small channels are the bile canaliculi, and then we have the bile ducts.
Bile ducts are the simple cuboidal?
Yes. So what epithelia would line our vessels? The branches of the portal vein and the hepatic artery. What kind of epithelia lines blood vessels? Simple squamous. So that's how we tell the difference between the 3 structures. Okay. Now before we look at the portal triad itself, let me explain a little bit further about these bile canaliculi. So if we have, now I erase the blood vessel sequence there, and I'll draw a couple of hepatocytes. Okay. So here would be an hepatocyte. They don't have microvilli. What they have are called microfolds or little rugae on each side. And then their back, kind of like the curvature, your lumbar curvature, the plasma membrane separates. This is not lined by any additional epithelium. It's just the plasma membrane of the hepatocyte where it's not fused together. And this is the bile canaliculus. So if I were to take 40 people and line them up back to back, you know, all of us have this curvature, you'd be able to pass a rope through that, all right? Between the two curvatures of each person's back, we would have this small tunnel created. And that's what the bile canaliculus is. And so bile is going to be found in that space, and it's going to flow in the opposite direction as the blood. It's going to flow out towards the corners of the lobules. So here and here on each side, we would have the simple squamous epithelium of a sinusoid. So each hepatocyte is facing a sinusoid. So it has ready access to retrieve any amino acids or glucose or lipids from the blood, or secrete into it any new proteins that it's making, or if it's releasing any glucose. Okay? So every cell, every hepatocyte has access to the sinusoids, and that's why they're never more than 2 cells thick. All right, so they can have that access. So we have these 3 structures. We have, and these are identified as the 3 structures that form a portal triad. So we have the branch of the portal vein. Again, these triads run up and form the corners, the 6 corners of a globule. We have a branch of the hepatic artery proper. And we have a bile duct. Not one of our named ducts, but these would be the tiny little ducts that would come together to form the left and right hepatic ducts. So this is from a pig. Human livers don't have this much connective tissue, so it's a little bit more difficult to find the portal triads, but they would exist at each of these corners, and they would be shared with another hepatic lobule. Okay? So when physiologists look at this, they actually create a range in which the cells on either side of this would have access, have the highest concentration of nutrients and oxygen. The cells closest to the central vein will have the lowest. There's physiological regions related to the diffusion of those components. Okay? So just another illustration of the central vein and the corners of the lobules. So as we look at this image, another one here. Here we go. So these bases here would be the sinusoids. You can kind of imagine that they're all kind of flowing towards one direction, and this would be the central vein. Notice the central vein doesn't appear to have very much connective tissue. Like the sinusoid just empty directly into it. All right? Whereas in this image of the triad, you can see a lot of connective tissue and several different structures. So this large structure here would be the branch of the portal vein. This structure here that has a smaller lumen, but about the same amount of tunica media, so a greater amount of tunica media versus the size of the lumen, that would be the branch of the hepatic artery proper. Notice all these nuclei here, it's a little bit like part of the macula densa. Those would be the simple cuboidal cells of the bile duct. Okay? Now, these other vessels here are probably lymphatics. We don't see any cells in any of them, so it's hard to say for sure, but they're probably lymphatics. So if we look at this model, and I'll bring it out in a moment, this would be the portal triad area. This would be the central vein. These would be the sinusoids going towards the central vein. Right? And the hepatocytes structure is lymphatic. That makes four things. Okay, so we talk about the portal triad, which is talking about those three vessels that I have listed there. So we have a couple of these liver histology models. They're made of plaster, not plastic, so they're going to be heavy. So don't drop them on your foot, you'll break a bone. So imagine if this is the central vein, over here by my left hand. This would be one of the 6 corners. So if I swing it around like that, this is a vertical cut through one of the lobules. Okay, so the bottom of the lobule would be down there, and the top of the lobule would be up near my elbow. So when we look at it here, this would be the portal triad, branch of the hepatic artery, branch of the hepatic portal vein, and the green structure would be the bile duct. Then these blue structures would be the sinusoid. Carrying the dark material inside would be the blood. And then over here, they're all the sinusoids are flowing into the center of the lobule. So we're just seeing the half, the radius of one of the lobules. This would be the central vein. The brown cells are the hepatocytes, and these green lines represent the bile canaliculi flowing towards the bile duct. Okay? So on part of the model, up here and down here, they kind of outline the simple squamous cells that form the sinusoids. However, there's another cell right there that's rather vague, and you probably wouldn't even notice it if I hadn't pointed it out. Okay, where is it? Over there. I'll hide it behind the top. Thank you. So, in the sinusoids is a cell. It's not, unlike the model, which is incorrect, it's not in between epithelial cells. This is a macrophage. So we saw macrophages in an unusual place in the alveoli, where they were on the surface of the simple squamous epithelium of the alveoli, protected from drying out by the surfactant. Here they're within the blood. And normally we find macrophages in connective tissue, all right? Because the blood's supposed to be sterile. We're not going to be expecting bacteria or viruses or foreign cells for the macrophages to break down. When we find those macrophages in the liver, they are known as Kupffer cells. So what are they there for? What are we breaking down in the liver? A normal component of our blood that we break down in the liver. Red blood cells. Exactly right. So it's the red blood cells, remember, which only live for about 4 months, 120 days. As they get older, their membrane becomes more fragile. They start to break down, and the macrophages engulf them and break down the proteins and recycle bilirubin, which is what makes the bile and the fecal matter kind of a yellowish brown color, and recycle the iron and the other amino acids back for making new red blood cells. Okay? So in the model, the Kupffer cell is shown right in here, which is incorrect. It's not a part of the wall of the sinusoid. It's actually within the vessel moving up and down through the sinusoids. Okay? Okay, I think we had a quiz question in here somewhere. Okay, question 3. Which of the following vessels forms the second bed of capillaries in the portal? Yes. Okay. One more time on number 3.
It's D. I literally left it kind of microscope.
Pardon? What do you mean by all?
Just the one.
There's only one.
Oh.
Yeah. So the complete
Yeah.
grammar of the question is which one of the following? triad means what number? 3, so you should have 3 answers. So you will probably see an image similar to this on our last quiz, which is Thursday. The quiz on Thursday will include structures of the colon, the liver, and the pancreas, and then 4 histology stuff, at least 4. So like recognizing like our first question, parotid gland tissue, or not parotid gland, salivary gland tissue. That wasn't necessarily the parotid gland. The recognizing by histology whether you're looking at the stomach or the small intestine or the rectum or the esophagus, paying attention primarily to the epithelia and its type and arrangement. Structures of the liver, pancreas, okay, so those components. The histology of the CPAP, so the muscularis mucosa, externa, and those components. Okay? So here's an electromicrograph of a close-up of the sinusoid here and the hepatocyte. All right. Sinusoids in the liver are fenestrated. Anybody remember what that word means? That's pores. So like the capillaries in the kidney. So this is why we can move the lipid material out, because the lipid, as we'll see on Thursday, is coated by a protein that allows it to be soluble in the water of the blood. And in fact, there's a small gap. So there's a simple squamous epithelium of the sinusoids and then the liver hepatocyte. And this gap in here is identified as the space of Disse, D-I-S-S-E. I think I have a larger view of it. So here would be the fenestra of the sinusoid, those microfolds I told you about, but all this empty space in here is that space of Disse. So there's a little bit of a gap space, interface between the capillary. Now notice all these black structures here? Those are too large to be ribosomes. That's what we learned the black dots were in Bio 430. But these are larger than ribosomes, which would be about that size. These are glycogen. That's how the liver stores the glucose, is as a starch, a polysaccharide called glycogen. It shows up in those black components. What 2 major types of organelles would you expect to see in hepatocytes? So I mentioned that they make lots of proteins. Those proteins are excreted or secreted from the liver cells. So what organelle do we use when we make proteins for export? Ribosomal endoplasmic reticulum. So the messenger RNA associated with the ribosome attaches to the RAR. All right, so we're going to have ribosomal ER and Golgi. Those go hand in hand. Ribosomal ER and Golgi apparatus, because they modify the proteins before they're actually leaving the cell. And then, what else does the liver do? This involves smooth endoplasmic reticulum. Smooth ER is utilized when the cells are making steroid hormones, which the liver doesn't produce. Every cell has some smooth ER for making new plasma membranes, but the liver has lots of it because it functions to detoxify. So it's going to take Tylenol and break it down. Acetaminophen, it's going to take alcohol and break it down. One of the first products of breaking down alcohol is glutaraldehyde, which is what we use to fix tissues so they don't rot. So if a lot of alcohol is being broken down, it can cross-link the proteins of the liver, and that can lead to cirrhosis of the liver. Infections or other damage to the liver can cause a lot of connective tissue. makes it difficult for the blood to flow through the sinusoids, and we get portal hypertension. We get a backup of fluid in the veins. Now, if the liver isn't working very well, it's not going to make albumin. So what are some of the signs we talked about? What would be a major sign that the body doesn't have enough albumin? Edema. Because albumin, as an osmotic protein, keeps fluid from leaking out of the capillaries due to hydrostatic tension. And so you get the swollen abdomen from ascites and swollen ankles from loss of fluid into the tissues because of the lack of the pulling back force of the albumin. Without the ability to break down the bilirubin from the red blood cells, that builds up in the liver as well. And that's why jaundice is a sign of liver damage. All right? Because that bilirubin leaks out of the capillaries and into connective tissue. So what do you think we're looking at here in this image of hepatocytes? The canaliculi. So this would be where two basement membranes are attached to each other via desmosomes. And this space right here, similar to what I drew in the upper right-hand corner over there, would be the bile canaliculus, a small segment where two cells have come together and the plasma membranes have not been joined by junctions. And that gap creates a small segment of the bile canaliculi. And again, all these black dots, glycogen.
What are the big black dots?
Microvilli.
Oh.
Oh, these?
Yeah.
That's mitochondria. Sorry, that's mitochondria.
Oh, those are mine.
Okay, this happens to be a liver with cancer. Now, in this case, the liver, you can remove part of something. The cancer is limited to one lobe. You can remove that lobe, and the rest of the liver will grow back. So in living liver donors, they don't take out the whole liver. In a kidney, they take out one kidney because you have two. All right. In the liver, they can take out a lobe or a partial lobe, and it will grow bigger in the recipient, and in the donor, it will return to normal size. For a while, for a few years, a lot of the surgeries for living liver donors were resulting in there was a high mortality rate, and so there was a ban placed on that, and only cadavers livers were used. That lasted for about 4 or 5 years, and I believe now, starting about 4 years ago, they're back to using living liver donors. All right, so we've looked at the bile structures here, so I'm going to leave this up and go back to our diagram and finish up with the pancreas before we take our break. And then it tails back to a point near the spleen. So this is known as the head of the pancreas. Most of the length is the body, and then it tapers off to the tail. And most of the pancreas is an exocrine organ, all right? Meaning that it secretes into ducts. So we have, similar to the salivary glands, we have this blackberry, mulberry style. It's like a pie here, but three-dimensional. We would have these pyramidal cells secreting into ducts. So these are known as acinar cells, similar to what we call them in the pancreas. And all those small ducts join together to pancreatic duct. Now the pancreas grows as an outpouching from the embryonic digestive tube. There's a right one and a left one. And then in development, the right one comes over and fuses to the left one. Notice the change of my hand here like this. So most of the left pancreas creates the main pancreatic duct, and then it flows into the right pancreas down this little dog leg. So this is the main pancreatic duct, as is this. All right, and the rest of the one from the left, that one that goes straight here, is known as the accessory. So there are actually two ducts that empty into the straight portion of the C shape of our duodenum. So this is the duodenum, where the common bile duct joins to the main pancreatic duct is known as the, the long name first, hepatopancreatic ampulla, or short ampulla of Vater. And that's the common channel through which both bile and pancreatic secretions will travel. Okay? As I mentioned, there is this sphincter, and that's called the sphincter of Oddi. Sounds like a Star Wars movie. Okay. And so that's shown here. And this empties into the duodenum at a slight rise. Now, this is way out of proportion. It's much smaller than that. But this slight rise is known as the major duodenal papilla. The accessory would empty into a minor. Right out the major. Okay? So it's just like a kind of a hill that rises up, in the middle of that would be the opening for the ampulla. So when we talk about digestion, as we get to the small intestine, remember I mentioned earlier on Thursday that we have three organs coming together with their contents in the duodenum. So we have the liver releasing the bile. We have the stomach releasing the churned food or chyme. And we have the pancreas releasing its juices. Okay? So we know the bile salts coming in from the liver. What would be the two major components from the pancreas? We'll talk about those after break. But anybody have an idea what they are? Yes. Right. So we have digestive enzymes. Those have to work before the small intestinal enzymes will work. And the bicarbonate ions we've been talking about since we started acid-base balance. Okay, so those are the two secretions of the exocrine part of the pancreas from the duct. Now, most of you are aware the pancreas also has an endocrine component. We'll talk about that on Thursday night. So these would be the islet cells, releasing primarily the two hormones insulin and glucagon. So we'll cover that on Thursday night. These would be all the acinar cells and their ducts that come together to form the main pancreatic duct. Okay. So the pancreas in histology looks most like the parotid salivary gland. And what's going to help you tell the difference, and I'll make sure it's there if I'm asking for pancreas and not there, obviously if it's the parotid salivary gland, would be these acinar cells. Okay? So that's going to be the distinctive factor that will let you know you're looking at the pancreas and not the parotid salivary.
Which one's the acinar cell?
The light ones,
Oh, okay, so both of the big.
all of these right here. Everything else mass around it would be the acinar cells of the exocrine component. And that's going to be secreting into blood vessels because it's hormones, not into ducts. Okay. All right, so let's stop there. Let me know, take a break and study. Let me know when you're ready to take the quiz. The quiz covers structures we started on Thursday, so starting with the mouth, teeth, through the stomach. Okay? So review your blood vessels, review your mesenteric structures, the patterns of the teeth, the GPAT. So even though it's not histology, I want you to know from our intestinal model. Here, not the villi and the lacteal or anything like that specifically from histology, but the mucosa, the muscularis mucosa, the submucosa, the muscularis externa with its inner circular and outer longitudinal, and the serosa. Okay, from the model, not from histology, not from tissue slides. All right.